Hey guys, welcome to Chapter 18, Topic 2. In Topic 2, we're going to talk all about the M phase, which is mitosis and cytokinesis. So here's our topic outline for this chapter, or for this topic. We're going to review the M phase, we're going to talk about the control mechanisms, and we're going to talk about some of the parts of it. As always, here's our topic objectives. These are the objectives I expect you to have mastered before you move on to the next chapter. So let's start with what is the M phase. The M phase includes mitosis and cytokinesis. So it's really important that you understand that M phase does not necessarily equal mitosis. M phase equals mitosis plus cytokinesis. It happens really quickly compared to the rest of interphase. As I said in the last topic lecture, the cell will mostly be in interphase for its life. The M phase is a very, very short period of time. And the goal at the end of the M phase is to create two identical daughter cells that were exactly the same as the parent cell. So it's really, really important. Now the focus of this is always to make sure that those chromosomes separate correctly. That is really the big key. If the chromosomes don't separate correctly, the daughter cells are not going to have the right chromosomes to function properly, and they're going to be missing some genes. So now let's look at how we move into M phase first. So at this point the cell has finished up G2 and we're ready to move on to M phase. All the correct signals are there, we're ready to go. So what you see here is the activation of the mCDK. So the mitotic CDK shows up, binds the M cyclin. In this case we have the inhibitory kinase and the activating kinase are going to show up and phosphorylate this mitotic CDK. You can see that here with this inhibitory phosphate. So once again, there's two inhibitory phosphates. The diagram just only shows you one. So once again, the complex gets phosphorylated in three places, but only one of those is actually an activating phosphate. The other two still inhibit. And it's not until the activating phosphatase removes the inhibitory phosphates that this becomes an active CDK. And then once this is activated, now we're ready to stimulate more activation and we're ready to move on to the next cell, or the next phase. And so we can see that here on the next picture is that the active mCDK actually creates a positive feedback loop to create more mCDK. So once we've fired this mCDK, once we've said go, we're going to divide, this is really an all or nothing process. We're going to charge right on through this. There's not really a whole lot of checkpoints after this. So it's really important that we understand that mCDK is going to create more and more and more active mCDK to drive us into M phase. So we talk a lot about how chromosomal division is really, really important. And part of this has to do with how do we condense those chromosomes, because remember they're really, really long and they're floating all over, well, they're in their spots in the nucleus. But if we left these balls of yarn undone, we would have some massive problems when we tried to pull them apart. So it's really important that we get them condensed down. And at the prophase, and at the first phase in M phase, prophase, this is the first place where we're going to be able to see them under a light microscope because they've condensed so much. And they condense through the use of these proteins known as condensins. And you can see here how they help get the chromosomes condensed down. So the cytoskeleton plays a huge role in M phase, and there's two main cytoskeleton machines you're going to see us talk about in the next couple slides. We have the mitotic spindles and the contractile ring. And we're going to talk about each of these a lot, so let's not worry about them too much, but I want you to make sure that you understand that that's what we have to deal with. So we have mitotic spindles and the contractile ring. Alright, so let's get started in M phase. We have 6 subphases to M phase. We have prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. Now, your book will go through how this process happens based on the movement of the mitotic spindles and how things are moving that way. I'm going to stick to this lecture keeping them in their phases just simply because that's how you've seen this material presented before, and that way we don't throw things totally out of context. But it's important that you understand that these phases are not necessarily their distinct events. In fact, they flow quickly from one to the other. They're separated out just for our well-being, and there's a couple defining hallmarks of each phase. But in cell division, they really move from one to the other on their own. And it's not really this clear division line. And so it's really important to understand that. But we're going to stick with the phases, but make sure you note that it's all about where these chromosomes and microtubules are moving and what they're doing. All right, so let's start with prophase. Prophase, we got our replicated chromosomes, we're condensed down, we're getting ready to get started. At this point, we still have the intact nuclear envelope. So these chromosomes are still kept somewhat located inside the cell. But what they have that's forming on them are these kinetochores. And these are protein complexes that form near the center of the protein, or of the DNA. And it's important because it's going to help us pull apart those chromosomes later. But right now, that kinetochore protein is just forming on the inside. What's also forming is the centrosomes. And so we have this... we have the centrosomes, and then we have the asters that are forming where the mitotic spindles are coming out. And these are going to start moving towards the poles. In pro-metaphase, we lose the nuclear envelope. And what this means is now that those mitotic spindles can connect to those kinetochores. And the microtubules are also going to then spread across the whole cell. So they're going to form the mitotic spindle. So you have two things. Because remember, the microtubules are subject to dynamic instability. If they're not connected to something, they're going to fall apart. And this is what happens. As the microtubules are shot across the cell, if they don't bind to either a kinetochore or to another microtubule, they're going to disassemble. And we're going to look at that here in just a second on this slide. So this just helps us keep track of what's happening so far with those microtubules. So far you see the asters are formed with the spindle pores on the centrosomes, and they've shot across the cell. The ones that join to other microtubules are known as interpole microtubules, and they form that mitotic spindle, and this is kind of a nice structure to help the cell process this whole situation. The ones that join to the chromosomes are known as kinetochore microtubules, and these are going to be used to help pull apart those chromosomes. All right, so in stage 3, metaphase. Metaphase is when the chromosomes are going to line up at that metaphysical plate. And so they're all lined up in the middle, and we've seen this all the time in lab and everything. You've seen this example many, many times, but they're all lined up, and they're getting ready to be pulled apart, but right now they're still attached. Now remember, the chromosomes have structures that are helping hold them together, and at this point they're going to start to be those structures that help pull them together are going to start being degraded so that they can be pulled apart when we get to the next phase. And that's anaphase, and this is where the microtubules are going to start to pull apart from each other so that the chromosomes are going to pull apart. Because remember, the key to this whole process is to ensure that we have equal number of chromosomes and the same type of chromosomes on each side. It wouldn't matter if we had 46 on both sides, but one side had 2 number 1s and one side had 2 number 2s. No, we need one of each. So we need to have all 46 be the correct on one side and all 46 be the correct on the other side. And so this is how anaphase starts to do that. So let's look a little bit closer at the two mechanisms of action that help this occur. And this anaphase A and anaphase B are occurring at the same time, so it's important to know that. So in anaphase A, the chromosomes are pulled towards the poles. So this happens as the microtubules are shortening, they're pulling them towards them. This is the action that we think about a lot. But anaphase B is also occurring and is also really important. This is when the poles are pushed and pulled apart from each other. So as you can see, the cell is even expanding. They're kind of pushing in opposite directions. And this is through the sliding force that's generated between those interpole molecules and helps pull those even farther apart to make sure that these chromosomes and everything else for the cell division stays even farther apart. Now in telophase, we see the formation of the contractile ring starting. You can see that on the little red nodules here. And so what's going to happen here in telophase is we've got those poles being pulled apart, the nuclear envelope is starting to reform, and the contractile ring. So we're almost finished with the M phase at this point. So the nuclear envelope, when it was disassembled, all of this parts stayed in place, but they just kind of got phosphorylated and they're still there, but everything is ready to go. What happens is at the end of telophase, all of these phosphorylated lamins and nuclear envelope vesicles and everything, they become dephosphorylated. And this encourages them to reform as a nuclear envelope. And so they come back together, reform the nuclear envelope, and that's what we're seeing happening in telophase. Now here's our last step of the M phase, and this is cytokinesis. And you can see the contractile ring is formed, and we can see the nuclear envelope is formed. The chromosomes are decondensing. The interphase microtubules are starting to go back in the centrosome. Everything's starting to look a lot more like interphase. And during this cytokinesis, this contractile ring is what's going to equally divide the two cells. And it's important that they're equally divided because we want them to be identical at the end. Now remember, this is only happening in animal cells. Plant cells will do cytokinesis in a different way because they have a cell wall. But this contractile ring is going to squeeze in the middle and help pinch these cells into two new cells. For plant cells, what we actually have is the formation of a new cell wall right smack in the middle of the two components. And this happens because, as I said, the cell wall, we can't pinch it off like we can in an animal cell. And it's used creating a series of the microtubules and some vesicles from the Golgi that then fuse together to create this cell wall. So it's important to understand that plant cells do not undergo cytokinesis the same as animal cells, but for the main focus of this class, we're really concerned about how the animal cells work because that's been our main focus of the entire class, has always been on eukaryotic animal cells. Alright, so this is the end of topic 2. So if you have any questions, please let me know. And when you're ready, you can move on to topic 3, which is programmed cell death.